Alright everyone, so in this video we're going to go over the third step in our central dogma, which is translation. So the central dogma again is DNA to RNA to protein, and translation is a step of converting RNA to protein. So leading off from transcription, we have our mRNA strand that we will then bring into the ribosome, and then tRNAs will bring in the amino acids that will link up to eventually make protein. And before you guys went over like ribosomal subunits, the 50S and the 30S make the 70S in the case of the prokaryotes. And the 50S would be the bigger ribosomal subunit seen here, and then this would be the 30S. When you bring them together, that makes the 70. So this is what those ribosomal subunits are in reference to. However, it doesn't really look like this. If we take a top-down look from it, it'll actually look like this, where we have the sites more in a triangular shape than of a linear shape. But just for visual purposes, we're just going to have it look like this. So after transcription, we have our mRNA strand here. That's going to go through the ribosome. Then the tRNA will read the mRNA and bring in the appropriate amino acids. And it reads that by using the anticodon, which has to match up with the codon of the mRNA. Now, getting a closer look at this, we can see that our ribosome is reading the mRNA, and we have our tRNA here, and at the bottom it has an anticodon. So that reads the codons on the mRNA. Then the tRNA, for any given amino acid based off of the mRNA, will come in, bring in the amino acid through the A site, which is called the amino acyl site, then go over to the P site, drop off the amino acid, and that's where all the amino acids will link out of, and they leave the P site. Afterwards, the tRNA carries a charge, so it doesn't go back to the A site, it goes next to the E site where it goes outside of the ribosome. We don't want to back up here because the next tRNA is bringing in the appropriate amino acid. So it's based off of charge. When the amino acid comes in with the tRNA, it goes into the A site, links up in the P site, then the charged tRNA has to go through the E site, that's where it's attracted, and then goes outside of the ribosome. And this, it's like a conveyor belt. It just keeps going and going until the stop codon on the mRNA says, that's it, that's all the amino acids that we need, our protein is done. So a question that Dr. Bazan likes to ask on his tests are, where do the amino acids leave out of? And that's the P site. And you guys might think, oh, this is an easy question. But that's the gist of it. He'll make it kind of wordy so that you'll think that he'll be asking about the tRNA. And sometimes he does. I'm not sure for your guys' test which one he's going to ask, whether it's going to be the amino acids or it's going to be the tRNA. But just read the question carefully and know that the amino acids go out of the P site while the tRNA leave out of the E site. So you have two things that are leaving here. Again, tRNA leaves out of the E site while the amino acids leave out of the P site. So just to go over, we're going to have our mRNA being fed through the ribosomes. As it's doing that, the tRNA is reading the mRNA with its anticodon in order to bring in the proper amino acids. So it does that by bringing them in through the amino acyl site. The amino acids will link up in the peptidyl site. Then the empty tRNA will leave out of the E site while the amino acids go out of the P site. And this same reference down here is showing you from the other view is that it just goes around and around. It's like a factory. Since prokaryotes don't have membrane-bound organelles, this all happens in the same place. So they go through transcription and translation in the same place. While eukaryotes, transcription happens in the DNA, the mRNA is sent out, and translation occurs in the cytosol. But there are two types of ribosomes that we look at with eukaryotes, and that is the free ribosomes, which are in the cytosol, and those are typically used to make proteins that are going to be used for the cell itself. while we have ribosomes that are in the endoplasmic reticulum. Those are proteins that are typically going to be made for other cells. So we make proteins and send them out. So that's the big difference between prokaryotic ribosomes and eukaryotic ribosomes. If you guys have any questions on this, please feel free to shoot me an email.